Hi everyone, this is going to be the last video for chapter 16. So this will be on covering enzymes, and I'll try to keep this as straightforward as I can. So why don't we get started? So enzymes are a type of protein. So the way you want to think of it is almost an umbrella term. All enzymes are proteins. Not all proteins are enzymes, right? We said some proteins are structural or they do other functions. Enzymes are biological catalysts, which means they make chemical reactions happen faster. Typically, you have this giant structure. We don't get into why proteins are so large. There's a reason for that. When you get into maybe higher level chemistry, it deals with entropy. But you have a big protein, but there's often a small spot on the surface in which the amino acids are arranged in such a way that they can actually bind a particular molecule and then do chemistry on it, right? And so that's what you're kind of seeing, the overall picture in this sketch right here. So why don't we go a little bit slower through it, if it lets me. There we go. So enzymes as biological catalysts, right? So they catalyze nearly all chemical reactions taking place in the body. So some reactions in the body can take up to it's a ridiculous amount of time. You're talking thousands of years for them to happen naturally, but your body can get them done in less than a second. The quickest a reaction happens, it's usually about 10 to the 8th reactions per second. That's about as fast as they can happen. That's very quickly. So you're talking about reactions that happen in less than a second. And that's the way that you can, when we operate, we operate on the unit of time of milliseconds, seconds, minutes, and hours. So things have to happen quickly for you to respond to the environment and for you to live, right? We've seen this diagram before about having an uncatalyzed reaction where there's a large activation energy. So there's a barrier that needs to be overcome. The reaction usually happens more slowly with these higher barriers. We mentioned catalysts in the past. This is more the high school definition. It's not my favorite, but it will work. We can say catalysts lower the activation energy. So given that the way that the protein and the compound that it reacts with, right, when they come together in such a way that actually takes less amount of energy for that reaction to occur. So it happens at a faster rate. In reality, it doesn't look like this picture. This is a really simplified picture. In reality, it usually happens in 2 subsequent steps or more, but those steps are usually lower energy than if they were uncatalyzed. And I've also talked about this before, naming enzymes. So if you hear a protein name and it ends in ACE, so ASE, that is usually talking about an enzyme. And so they usually name it on the substrate that it acts on. So a substrate is the molecule that the enzyme reacts with. So if your substrate is sucrose, that means the name of the enzyme that works on it is going to be a sucrase, right? If you're doing an oxidation reduction reaction, you're probably going to be dealing with an oxidase. So that would be something that takes it in the oxidized direction. So it's losing electrons. If something is gaining electrons, the substrate, you'll probably call it a reductase. If it does both, you call it an oxioreductase, right? The only thing that's a little bit different deals with digestion enzymes. They do add an end in IN. So like pepsin is found in the stomach that can break down protein bonds, and the same thing with trypsin or chymotrypsin. I'm not too worried about classifications. You kind of pick up on the names. They're actually pretty straightforward. Biologists, it's funny. In some senses, biochemists and biologists are really boring with naming things. In other times, they're really really weird names, right? So the boring ones. So if you're doing oxidation reduction, oxioreductase. If you're moving things around, you're a transferase. If you're conducting a hydrolysis reaction, so adding water across a double bond, you're a hydrolase. So you're literally adding water. If you're moving or adding or removing atoms, you're a lyase. If you're rearranging things in space, right? So we're talking about isomers, they're isomerases. And if you're actually attaching something, right, you're going to be a ligase. Typically, you're adding ATP, or you're using ATP. Typically, you're adding like a phosphate group or something like that would be a ligase. So you're adding something to it. This is a little bit of a side because these are the boring names. I kind of looked up some of the crazy names that biologists come up with. It's almost like people get bored in lab. So like biologists are great at making up acronyms. So and this actually plays relevance to us in the news. So if you were hearing about the tests for COVID-19, you might have heard something called an ELISA assay. And ELISA just stands for an enzyme-linked immunoabsorbent assay. The Z gets thrown in there from the immunoabsorbent, even though it's an S, but it's spelled ELISA, or something like FRET. That's fluorescence resonance energy transfer, or my favorite one that I used to work on these class of proteins called SIMIBI, signal recognized particle MIN-D and bio-D type nucleoside triphosphate binding proteins. So the names are really detailed, but sometimes we give them dumb names because it's fun. It honestly comes down to that sometimes. This is a funny anecdote, and you can skip if you don't want to hear it, but there are such things as genes and proteins called sonic hedgehog genes. So they're called SHH genes now, but they stand for sonic hedgehog. The reason for that, it was in the 1980s, and Sonic Hedgehog had just come out, and it basically is a mutation in fruit flies that made certain structures look like they had spiky proteins. So they called them Sonic Hedgehog genes. Someone tried to name a protein after Pikachu once because it dealt with transferring electrons like electricity. Nintendo threatened to sue. And then biologists and biochemists realized we need to be a little bit more structured in our approach of naming things. So Pikachu genes don't exist, partly because Nintendo threatened to sue, and then partly because people started to realize that if you have mutations in certain genes and you can never tell where a disease is going to come from, you don't want to have a doctor sit someone down and say, you have a mutation in your Pikachu gene and you're going to die. So that's part of the reason why some of the names have become a little less flamboyant, we'll say. All right, enough of that. Let's get back to science. So how do proteins or enzymes specifically do their chemistry, right? So it's not magic. The proteins need to collide with their substrate or whatever molecule they're acting on. It can be a molecule. It can also be another protein. And when that substrate binds to the active site of the enzyme, chemistry happens. Sometimes it's quick, sometimes it's slow. But then after the chemistry happens, the product is released. And that's the same term that we use when we're dealing with chemical reactions. It's the resulting chemicals. They're released from the active site and they basically let go and they float away. So the chemistry has been done. So in an enzyme catalyzed reaction, right? So it's a stepwise process. First, the substrate attaches to the active site. And so with that, chemists and biochemists typically use it as E for enzyme and S for substrate. And when they bind, we call it the ES complex. And then we have the double-headed arrow saying that it's an equilibrium. It's not a covalent bond. So they come into contact and sometimes they release, sometimes it stays there. It's statistically determined. We're not going to get into that. It's called something called the dissociation constant. It does dictate how strongly it binds. It also dictates why certain medicines work and some others don't. Once the reaction occurs, it's usually unidirectional. Not saying that it can't be in reverse. There's plenty of enzymes that do it in reverse. And actually, theoretically, all enzymes can run in reverse. But based on how your body functions, typically it's unidirectional. The enzyme's regenerated. That's why we call it a catalyst, because a catalyst always is never consumed. It is always regenerated, and it releases product. So that is the most general, and it's the canonical way that enzymes react. Enzyme and substrate form ES, chemistry happens, and E plus product, and product is released. So how does this happen? And we have good ideas, right? We actually have pictures on the molecular scale of how this happens, but we don't know, we have theories about the process of how it happens. We've modeled it in computers. We're trying to see it in real life. But we still operate under models. And so the first is lock and key, which you might have heard in biology before. It's the idea that the active site is rigid and non-flexible, which I can give you plenty of examples where that's not true. But again, remember, it's a theory. It's called first assumptions. This is what I'm going to start with. If things get harder, I got to fix my model. But you start here. Lock and key, rigid model. It works for some proteins, not a lot. Actually, no, it does work for a lot. I'll correct myself, but it's not 100% true, but it's a good start. It's what I was saying before, right? Proteins are constantly vibrating, so that active site is constantly changing shape. It really depends on how accessible that active site is. Maybe the substrate doesn't care when it's shaped like that much. Maybe it does a lot, right? It affects how fast the enzyme reacts. So usually what happens, if you're looking on a big, whole forest view instead of the trees view, it's just basically saying that the enzyme's active site is a perfect fit for the substrate. Perfect is a little bit probably overboard, but a good enough fit that it can distinguish between different types of reactants. It will only work for certain ones, right? So it's the idea of a lock fitting in, key fits into a lock type thing. So it's saying it's static, which we absolutely know is not true. Proteins are not static, which means they stay still, right? But there is, it's not including flexibility in the structure. In real life, there's flexibility. So again, it's a model, and that's why it's not the only thing that we say is true. Another one is the induced fit model. This is getting a little bit more towards reality. It's saying that like the enzyme active site is mostly designed for its substrate, and then it's kind of vibrating, and the substrate itself is vibrating and moving in shape, right? They don't move too far, but enough. And then basically when the substrate starts to bind to the active site, they both take on a shape that's mutually beneficial for that to happen. And so basically that's why it's induced fit. So the fit's not perfect until they come together, but the interaction of the two causes the perfect fit, which then leads to catalysis. That's more leaning towards what is correct. Again, models are models, right? Not every enzyme is going to follow this model, but induced fit in lock and key kind of take them together. But as far as we know, right, the enzymes have they only going to react with certain things. You can actually get them to go off, you can make them do reactions they're not meant to, but they really do only try to react with what they're made for. So what are things that affect enzyme activity, right? So number one, this is why humans die of fever if you get your fever too high. Enzymes are only designed to operate at a certain temperature, usually around 37, though it's plus or minus a little bit. Once you get over 50, human proteins pretty much are done, but at 50 degrees, you'd definitely be dead if your body temperature was that high. But they do precipitously fall off in terms of activity when they get too hot. The same idea, if you get hypothermic, your proteins aren't moving enough. So literally they're designed to vibrate in a certain way so that they react in a certain way. If they get too hot, so above 37, sometimes they literally can shake themselves apart. And then they die, literally, we call it dead. But the protein has denatured itself and it no longer functions. If it's too cold, they don't vibrate enough and that induced fit model doesn't fit, and then they stop working. So humans are designed, and animals and plants are designed to work in a very narrow range of temperatures. And the reason for that is because enzymes are they were evolved to work only in narrow temperatures. Also pH and enzyme activity. And you might have heard this in terms of COVID-19. Right now, one of the theories is that it's changed the pH of cells. And the same thing with remdesivir, and the same thing with hydrochloroquine. But remdesivir is now just almost, and I don't know if it's been yet, maybe when it's published, been approved by the FDA for treating COVID-19. It can affect the pH of cells. Well, why would that matter? Because certain R groups are affected by the pH. Even just small changes in pH, even if it's not super acidic or super basic, you can start to change if something has, an ammonium group or an amine. Maybe it's carboxylate, maybe it's carboxylic acid. It really can affect, and if you basically, let me just draw this out real quick. Right, a carboxylate group can take a hydrogen and that can do some acid-base chemistry. If somehow you drop the pH enough and it's protonated, it can't do its chemistry anymore. It's literally jammed up and it's done. And so enzymes, again, have evolved to work at a certain pH. So most enzymes, like the book is saying around 7, that's a huge generic. That's like most cells. But I mean, like inside your intestines can get to pH 10. Your enzymes like pepsin and stuff inside in your stomach operate at pH 2, right? Or 1. It's a generic number, but 7 is usually what they operate at. And again, if you get them too high or way too high and too basic or too acidic, you literally rip the proteins apart because you're breaking the amide bonds. Other ways are actually how medicines work, are usually inhibitory molecules. So we've already talked about how enzymes can catalyze things. So an enzyme and a substrate come together, they're in equilibrium, they form an ES complex or enzyme substrate. Chemistry happens, the enzymes are generated and the product's released. Sometimes you can add something called an inhibitor, which is usually when we talk generically about drugs are inhibitors. They also bind the enzyme, they bind it up and essentially don't let go, or that they do let go, there's so much around that it just gums up the work and the protein can no longer make product. And there's going to be 3 types of inhibitors that we look at. The first is going to be competitive, which literally means that the enzyme itself, if that's the active site, I'm going to make that little indent there, the active site, right? If the substrate, if that's where it binds, sometimes the inhibitor literally binds to the same spot, but it's designed in such a way that chemistry cannot happen. So what it does is it literally gums up the works. The substrate cannot get in, and so no chemistry happens. So it literally, if it gets shuttled, it's called shunting, but it gets shuttled off or shunted to the EI complex or enzyme inhibitory complex. There's no way you can get substrate in there. The enzyme is effectively dead until that inhibitor is cleared away. And so that's how most drugs work, not all drugs, but that's one mechanism of action. Another one is, this is the same picture, right? So if the active site is designed like this, the substrate binds, you have your induced fit. So you can see how the enzyme and the substrate both change shape and the product's released. If you have competition with an inhibitor, well, there's my same active site. The inhibitor is slightly different, but it's good enough to bind it. Well, it sits there, the substrate can't do anything. It just floats around and nothing happens. Non-competitive inhibitors, we can go through this, you can read through it, but sometimes pictures say a lot more than words. We have our generic enzyme structure. Again, substrate would bind to the active site. Sometimes what happens is an inhibitor binds somewhere else on the enzyme. Maybe it's somewhere on the outside. And what it does is it changes the shape or into the active site into a different shape. So the substrate can't fit in there, and then it no longer binds. So in the sense it's non-competitive is that they're not going for the same spot. It's just binding somewhere else. The book doesn't get into it, but it's called allostery. Just means somewhere else is affecting the active site. So what does that look like in the same model we had before, right? We have our active site. If there's no inhibitor, substrate goes in, product comes out, nothing new. The inhibitor now doesn't bind to the active site. It binds somewhere else on the enzyme, changes the shape. Well, the substrate just floats around and does nothing. It no longer can do chemistry, so it's effectively dead. It's another common way of making drugs. The last one is an irreversible inhibitor. These are actually the best type of drugs you can design, but they're very difficult to design. Usually they're called warheads because when it hits, the entire protein is knocked out for good until your body eats it. What it does is it usually makes a covalent interaction. So it literally is irreversible. There's no way to knock that inhibitor off because both with competitive and non-competitive, theoretically and statistically, you can kick it off by giving it a boatload of substrate. We're not going to get into statistics of how that happens, but they're not covalently bonded. They're just attracted to each other and they change the shape. Irreversible inhibitors, colloquially called warheads, they literally bind somewhere on the enzyme and just kill it dead. Again, these are the best type of drugs because they cannot be undone. But also, if they're designed to kill you, then there's no way to undo them either, right? That's why they could also be toxic because they form covalent attacks. So nerve gas, insecticides, occasionally antibiotics, though usually they don't have to be. Some are. Antibiotics have a huge range of what they do, so we won't get into that. So there's a little bit summary. I'm going to skip over that because you guys have those slides. I know I've talked a bunch today, but we are done now with chapter 16. So I know that was a lot. It's everything that we talked about. I think, again, this chapter 16 is probably the most important chapter we've talked about probably since the last time I saw you guys in class. So, all right, we are done for now with chapter 16. If I can find how to stop this. There we go.